Hello friends, welcome in another video lecture of EG Life Science. Here I am talking about antigen presentation by a MHC second molecule, a exogenous pathway or a vesicular pathway. So please subscribe my channel EG Life Science, like my video and share it to your friends, and don't forget to press the bell icon so you will get a notification of my newly uploaded video. So let's start. By a endocytic pathway, the antigen presenting cells present or shows a antigen or a processed antigen to the CD4 plus cell or a T helper cell. In this pathway, the antigen is outside the cell, so this antigen is called as a exogenous antigen, and it is processed within a cytoplasm, so it is called as a endocytic pathway or a vesicular pathway. The APC, like macrophages, dendritic cell or a B cell, can internalize antigen by a phagocytosis, endocytosis or a both. Macrophages internalize antigen by both process, whereas other APC like dendritic cell and B cell are not phagocytic, or they are poorly phagocytic. Therefore, they internalize exogenous antigen only by a endocytosis, either receptor mediated endocytosis or a pinocytosis. The number of events occur in this processing. The first event, peptides are generated from an internalized molecule in endocytic vesicles. Once an antigen or a peptide antigen is internalized, it is degraded into a peptides within a compartments of the endocytic processing pathway. For processing, they requires 1 to 3 hours, and after it, the peptide is ready to present with a class second molecule to the CD4 cells. The endocytic pathway appears to involve 3 increasingly acidic compartments: a early endosome having a pH 6 to 6.5, a late endosome or a endolysosome having a pH 5 to 6, lysosome having a pH 4.5 to 5. These three are the vesicles. The internalized antigen moves from early to late endosomes and finally to a lysosome. And in lysosome, there is a presence of a low pH and a number of hydrolytic enzymes. And these hydrolytic enzymes are hydrolases the larger peptides into a smaller fragments of the peptides. They includes proteases, nucleases, glycosides, lipases, phospholipases and phosphatases. Within the compartments of the endocytic pathway, the antigen is degraded into oligopeptides of a size 13 to 18 amino acids, and this is an optimum size for binding to a class second molecule. The early endosome, it moves periphery to the inward and become a late endosome and finally a lysosome. A small transport vesicle may carry an antigen from one compartment to a next, and eventually the endocytic compartment or a portion of them return to the periphery, and from cell periphery it fused to a plasma membrane, and in this way the receptor or these vesicles are recycled. Then next, that is the invariant chain guides a transport of class second molecule in endocytic vesicle. The class second molecule are synthesized within a rough endoplasmic reticulum, as well as the another protein also formed in a rough endoplasmic reticulum called as a invariant chain protein, and this invariant chain protein is a trimer protein. It consists of 3 pairs of class second alpha beta chains, and this trimeric protein interacts with a peptide binding cleft of a class second molecule. This invariant chain protein or this trimeric protein interacts with a peptide binding groove on a class second molecule, and this binding prevents any endogenously derived peptides from binding to the cleft, while a class second molecule is within a rough endoplasmic reticulum. The invariant chain also appears to be involved in a folding of a class second alpha and beta chains. Also, this invariant chain is responsible for exist of a class second molecule from a rough endoplasmic reticulum. This also routing a class second molecule to a endocytic processing pathway from trans-Golgi network to the cell membrane and on a surface of a APC. The invariant chain contains sorting signals in its cytoplasmic tail, and these signals directs the transport of a class second complex from trans-Golgi network to a endocytic compartment. Then next step or next event, the peptide assemble with a class second molecule by displacing a clip. A short fragment of invariant chain terminates a clip. The clip is nothing but a class second associated invariant chain peptide, and this clip remains bound to the class second molecule after the invariant chain has been cleaved within a endosomal compartment. The clip, a class second associated invariant chain peptide, this clip physically occupy the peptide binding groove of a class second molecule, and this prevent a premature binding of an antigenic peptide. A non-classical class second molecule, that is a HLA-DM, is required to catalyze the exchange of a clip with an antigenic peptide. For maintaining the structure and stability of a class second molecule, the peptide binding is required. Once the peptide antigen is bound to the class second molecule, a complex is formed, a complex of class second molecule with a peptide antigen, and this complex is transported to a plasma membrane where there is a neutral pH, and this neutral pH enables the complex to assume a compact and a stable form. The peptide is bound so strongly in this compact form, and it is difficult to replace with other peptides. Here is a representation or a schematic representation by a class second molecule. Exogenous antigen is internalized through an endocytic vesicle. Uptake of extracellular protein into a vesicular compartment of antigen presenting cell. Then it moves into an endosome. Simultaneously, in a rough endoplasmic reticulum, with the help of chaperone, the MHC class second molecule is assembled. It is made up of 2 chains of alpha and beta domains, and after complete assembly of a class second molecule in a rough endoplasmic reticulum, the peptide binding groove of this second molecule is occupied by a invariant chain protein, and invariant chain protein with a class second molecule is released from a AR to a Golgi, and Golgi to a endocytic vesicle. The endocytic vesicle and a endosome. In a endosome already there is a fragments of a peptide antigen. The endosome and a endocytic vesicle get overlapped to each other and forms a one larger vesicle, and the event chain replaced by a clip, the portion of clip is further replaced by a antigen, and a complex of a class second molecule and a peptide antigen is formed, and it gets transported through a Golgi network to the cell surface and presented to a CD4 plus cell or a T helper cell. The CD4 cell has a co-receptor and they binds with a alpha 2 domain of a class second molecule, and in this way they presents a antigen to a T helper cell, and then this CD4 cell takes a help of other cell to kill the antigen. This is a pathway which is presented by a MHC class second molecule. So thank you for watching my video. Please subscribe my channel Easy Life Science, like my video and share it to your friends.